Okay.
My group.
All right. So for the people that just came in, we're just getting started. We have the chat open. If you have any questions or if we're not being loud enough, you can tell us there and we'll be watching that.
Oh yeah. So wait, do you want to move that thing?
I don't know how far we could,
No,
yeah.
I meant the names, just so I can see.
Oh, yeah, good call, sorry.
Okay, so to start off, we'll be talking about protein trafficking. And so this topic covers in general how or what happens to proteins after they are produced in the cell, and how they can be moved around within the cell or outside of it in order to fulfill or execute their what they are supposed to do. So eukaryotes use a signaling system involving amino acid sequences within protein in order to signal where to move the protein around the cell. And we'll have like corresponding proteins that will, or molecules that will bind to the signal sequence in order to bring it to the desired place.
So we have a little flow chart up here, like just stand, sorry. So basically what this is covering is just like the central dogma. And I'm sure that's been like beat into your brains by now, but just all that is basically the path that you take from DNA to a protein. So you start off with your DNA in your nucleus, that is transcribed into RNA, which is then processed into your mature mRNA. And then that is translated into your protein. So protein synthesis starts in the cytosol on a ribosome where the mRNA will bind and then be translated. And after that, your protein, depending on whether or not it has a localization sequence and what its localization sequence is, can either be transported to the endomembrane system, which is like its own enclosed system. It includes the ER, the smooth ER and the rough ER, the Golgi apparatus and lysosomes, stuff like that. And then just all of the other stuff like the mitochondria, the nucleus. And because those are not a part of the endomembrane system, you can't have any like transportation of proteins between the ER and the mitochondria or the Golgi and the nucleus, but things within the endomembrane system can travel in one direction through the endomembrane system.
So the endomembrane system is a series of places in which a protein will go after it is recognized to have a signal sequence on it. And so what will happen is as the mRNA for a particular protein begins to be translated, at the very beginning, once the first few codons are translated, it will contain, if it has a ER localization signal sequence, it will have that at the beginning, at the N terminus. And so while it's being translated, it will have another protein recognize that signal sequence, and it will move the whole complex to the edge of the rough ER. And so the rest of the protein will be translated into the rough ER. And then after that, if it has no other signal sequences, it'll move through what is called the endomembrane system, the rest of it at least, where it'll go through a transport vesicle. It will pinch off and head over to the Golgi body. And then after going through there and being processed, if it has no other sequences, it'll head out of the cell through another transport vesicle. Or if it has a retention signal sequence, it can stay at the rough ER or the Golgi body or lysosome, depending on where it has a signal for it. But once it enters this endomembrane system, remember that it can't leave.
Right. Okay. So just a little more about that. So what Dave just said, you are not going to have functional proteins leaving the endomembrane. So what happens is, like, once a protein enters the endomembrane system, if it has a retention sequence, then it can stay there. If it doesn't, then it'll just move all the way through and it will be secreted outside of the cell. The only time that you're going to have a protein coming back out is like if it's misfolded or damaged or unfolded, and then you're going to have like your chaperones bringing it back out to go to the proteasome. So you're never going to have a functional protein thing, basically. It's a one-way street. And what I just said with that, you can have proteins be retained or secreted. And if they're going to be retained, they need to have a retention sequence. Otherwise, they're just going to go straight through. It's like one of those like moving walkways at the airport. Like once you're on it, unless you have something there to specifically say, all right, we're getting off. And then that keeps you, you know, like in the Golgi or in the ER, but you need something there, otherwise it'll move through and be secreted outside of the cell.
Okay, so here's a visual representation of the whole like protein localization process. So to start, a protein is being translated in the cytosol, but if during that process of translation, it has a signal sequence that is an ER localization one, meaning it will be recognized and brought to the ER. It'll go through this endomembrane system. But for all other signal sequences that we're looking at, they are recognized after the mRNA has been completely translated into the protein. And so there's a whole variety of places where these proteins can end up. And so for these three right here, for mitochondria, peroxisome, and chloroplast, if the protein has a signal sequence that determines that it's going to end up in one of these three, before it folds, a protein will come and bind to the signal sequence on the protein and guide it over to these, whatever the corresponding organelle. And once it goes in, just like the endomembrane system, for these three, it won't come back out. And then the protein will finish folding in here. The reason why is because the pores in these organelles are not big enough to have a whole folded protein go through. On the other hand, for nucleus bound protein, it'll have what's called an NLS sequence on it. And when the protein is, after the protein is folded though, that's when it'll be sent and like recognized by the important, it'll bind to the NLS and it will go into the nucleus after being fully folded. And unlike these three organelles and the endomembrane system, a protein that goes into the nucleus can come back out through like the export cycle. So a protein can have both an NLS sequence and an NES sequence. An NES sequence being one that means it's destined for export of the nucleus. And so here's like another diagram showing the possible places that a protein can end up in the endomembrane system by like going through first the ER and the Golgi apparatus, which is sort of like a post office where it'll like modify proteins and then send them off to like wherever else they need to go, depending on what other signals they have.
Does co-translational transport mean because it's going from the endomembrane system and the Golgi apparatus, and that one is specific?
Okay, so
Or is it
code,
like that?
code, yeah. That's a good question. So co-translational transport is because you know about the process about how like the whole process of translation where you have like this code, this mRNA code, nucleotides are like corresponding to codons, which are then this is being translated into a protein, right?
Right.
During that process, like while the mRNA is still in that complex, when like the first few codons go through and they're translated into like this into these codons, like the codons will be representing a signal sequence of signaling that it needs to be moved to the ER to finish up translation, right? Like that whole process, it's in the middle of that process when it gets transported
Oh.
to the edge of the ER, right?
Okay, yeah.
Whereas with these ones, like that whole process carries out, and then the rest of like this happens, right?
Right.
That makes sense?
Yeah.
So here is just like a different visual of that, and this shows kind of the difference between like how your protein would fold and enter your nucleus, and then the difference primarily between that and the ER is for the ER, your ribosome is going to be docked on a transmembrane protein. So your mRNA transcript is going to be translated by the ribosome, and then the ribosome is essentially going to thread it through this transmembrane protein like right away, and it will be folded inside of the ER. So, and then it can go to like any of these places where by contrast, like particularly if you're going into the nucleus, this is all going to happen in the cytosol. You're going to have your RNA transcript, it's going to be translated, then your protein is going to fold in the cytosol, it's going to move through the nuclear pore complex, or like what Gabe just explained would happen with like mitochondria, chloroplasts, peroxisomes. So the big difference that you're going to want to like remember is like with your rough ER, it's going to be studded with ribosomes, and that's because they're threading your protein as it's being translated into the ER to allow it to fold inside. And once you get inside, the ER localization sequence is going to be cleaved off. And it'll be processed. We'll talk about. Does that make sense?
So this slide is just like another like it's just a 3D rendering of like what we are going through, where it's like it just doesn't show like a free protein being translated into the endomembrane system. So this is just like the entire endomembrane system shown in like a visually appealing way, where you've got like this is like already the point where there's like these proteins have already been folded and translated into the ER here, and then they're sent off through vesicles to the Golgi body, and then sent off either to be secreted or to be central licensing. And again, that's if it has an ER localization sequence, and then it'll leave the cell if there's no retention sequences to keep it in any of these.
Just gonna check the chat real quick because it says there are two.
Oh,
Oh,
I cannot hear
no.
anything.
That's not good. Oh, it was a direct message, so I didn't see it on this computer.
Are we on mute or
No, we're not muted.
I think maybe when you're standing over there.
Yeah. Can we use these microphones?
Oh, I don't know.
Or does this only go to here? You guys hear us? It's really sensitive.
That's a bummer.
That was literally in my head. I'm going to watch the recording again.
Hey, okay, so if like our first 15 minutes or whatever, there's no audio, which would stink. Like you can always, we're gonna email Office PowerPoint, and if there's anything that you want reiterated, you can bring it to Office Hours. You can email any of us, but particularly the two of us.
Okay.
So,
Yeah, I have office hours.
okay, perfect, yeah.
Okay, great.
Mine are on Friday from
okay.
12:30 to 1:30, so.
Can people on Zoom hear me now? Gianna, can you guys hear us?
Okay.
How did you guys hear us before?
Huh, so some of the messages, okay, some of them are just showing up on this computer and some of them are just showing up on this computer, which does not make a lot of sense to me, but. Okay. Can you hear both of us?
Sorry, that's my leg shaking.
Wait,
No, no, you're good.
Did one person say they could hear us, but another said they couldn't?
I don't know what's happening with the chat right now. Okay, Gianna said that she couldn't hear anything. Carla said that she could, so. Until we are told otherwise, I'm going to assume that,
Why don't you just use the mic and then?
okay. Gianna, I'm not sure if that's just on your end, because we have somebody else saying that they can hear, so. Oh, if she can't hear me. The other people that can hear, so I think it might be on your end. But if it doesn't, oh, it's, oh.
You can just see if they can hear you up here.
Oh, can you? Sorry for the little intermission. If you want to take a look at your problem sets and figure out if you have any questions.
Yeah.
Those are big ones.
Oh, okay, she hears it now. Okay, all right.
I mean, it's never.
Really ghastly.
Okay, can you hear us without the microphone?
I have a.
Yeah, me, oh my god, like I'm jamming.
Can you hear us from over here? That's not the microphone.
When I did it with Van, I was like yelling at the computer over there and it is audible. So I think we're safe. Yeah, okay. I'm just gonna keep screaming.
Okay.
Okay. All right. Sorry for like the brief interruption there. I think we're sorted. So are you good with that slide? Gabe?
Yeah.
Yeah, okay. All right.
These ones don't.
Huh?
Nothing, I just, the slide was just. I don't know.
Just sort of stole the order that we were in.
Yeah.
Okay, so this is kind of just a reiteration of what we've already talked about. But, so what I was saying before, like, we can go from our free ribosome to the cytosol, and that's where it will stay if we don't have a localization sequence, or the mitochondria, or the nucleus, and those are all outside of the endomembrane system. So again, that means that you can't like go from the mitochondria to anything in the endomembrane. It's a totally separate thing. It's a totally separate network. And I think the diagram that Gabe showed kind of like reiterates that there's a very clear path that you move through. You can't cross your wires between endomembrane organelles and the other stuff. So this is the other stuff. And we won't talk about chloroplasts, but I'm not sure. I'm not a bio major. You might talk about it in like the plant bio class for bio majors. So.
Wait, I have a question.
Yeah.
Why is this like circle?
These are the ones that we're going to talk about.
Oh, okay, sorry.
No, that's totally fine. And then the chloroplast.
Okay.
Just like, you know that it's definitely know that it's not a part of the endomembrane system, but you don't need to know the specifics.
Okay.
Just like,
So none of these are part of the endomembrane.
none of these are a part of the
Okay,
endomembrane system.
Okay.
And I mean, the cytosol isn't really part of like any system. It's not
Right.
now, but
Yeah.
Yeah. So.
So signal sequences are usually embedded into the amino acid sequence of a protein. They're sort of like a unique zip code that tells where the protein destination will be. So these are not added afterwards, they're included in the mRNA coding for, and they are binding sites for other proteins to interact with. So you could think about this in relation to like the idea of KD and affinity. And so like these binding sites will have like affinity, a high affinity with the proteins that help traffic them to wherever they need to go. Because they need to be able to bind to them. And then, right, in terms of reversibility, like I mentioned before, ER localization sequences are not reversible. Once a protein is put into the endomembrane system, it's not going to come back out. Same thing goes for mitochondrial localization sequence. Once it goes into mitochondria, it's not coming out. Whereas for the nuclear localization sequence and export ones, those ones are reversible. And then, yeah, the retention signal sequences will be recognized after the localization.
When you say nuclear localization sequence, is that like in and out of the nucleus?
Yeah.
Okay.
Nuclear localization sequence, or NLS, is the sequence recognized when a protein, I mean, a protein can have it in the nucleus, but usually it's like when it's outside the nucleus,
Right.
it'll be recognized by important, which will,
It can go inside and then it can also leave it if it wants.
yeah, because it's because like these, it's because the fully folded protein can it can go in and out of the nucleus,
Right.
right? Whereas these other ones, like they're being translated in like single file, or in the mitochondrial case, like it's already translated and it's going in like single file into the mitochondria. So once it's folded, it can't come back out.
Right.
And then the other thing for localization is for ER localization is that once the protein is translated into the ER localization sequence is actually like chopped off from the protein.
Just to add on to your question a little bit there, you can also have a start sequence, so that will help you like that. So you can have like both. And so it can go.
Could I ask one question?
Yeah.
When how you said that ER localization sequence was like chopped off,
Mhm.
so does that mean all the sequences kind of stay on for everyone else? Or-
But yeah, they do, especially for the nucleus, because you think like, and we're gonna talk about this with import and export,
Okay.
but like if you have a protein that goes into the nucleus, then it needs its import sequence, it comes back out, it's exported, then it's in the cytosol. If it needs to go back in again, it still needs the nuclear localization sequence. And it would need the NES to be exported back out. You can think of like them having the option at least to move cyclically in and out, so you need those both at all times.
Right.
Okay. So this is just kind of a summary of signal sequences. You don't need to know the specifics of this at all. You just kind of need to know the trends. So I think the really important one to know is ER, you have your signal sequence really close to your N terminus. And the reason that that's important is because if for whatever reason you had a protein, and you probably wouldn't, but if you had a protein that had an ER import sequence and an NLS, because this is right at the N terminus, when it's being translated and read, that is going to be encountered first, essentially. So it's going to go straight to the ER, and that's like as the protein is still being translated, right? Because you're going to have your ER docked over that transmembrane protein, and it's going to be put in, threaded through into the ER as soon as that's read. So by the time that you get to like your NLS, then it's already gonna be in the ER like holding up, because this is kind of in the middle of the amino acid sequence. So know the trends, like retention sequence, that's closer to your C terminus, which doesn't matter because at that point you'll already be in the ER. And then peroxisomes are a little bit different, like it's always going to be specifically this sequence. And then we have our charged amino acids that are blue, or negatively charged in blue, positively charged in green, and our ER one is really hydrophobic. So just like generally kind of know those trends, but you don't have to obviously like don't memorize this. Just know if you were given on a problem like, oh, you have these two signal sequences, where would the protein go? Like be able to answer that kind of question.
So the so the ER and ER localization and retention sequences are examples of endomembrane signal sequences. And so for the localization sequence, before I was mentioning that there's like a protein to move on to, and so what the specific thing is SRP, which is a signal recognition particle, and that will bind to the complex and in the sequence itself, and will move the whole like ribosome to the edge of the ER. And without this, without a retention signal sequence, after it's been put into the ER, it will just end up going through the entire endomembrane system, like I showed before, which is the end stop for that is the outside of the cell.
Kind of just some more about like how your localization sequences work. So for like mitochondrial or ER or whatever, essentially your localization sequence is a sequence of amino acids, right? So what that's going to do is it's going to affect the folded protein's chemistry in a way that creates a binding site for a carrier protein. So your carrier protein is going to find this localization sequence, it's going to bind to the protein, and then it'll bring it to like the mitochondria or the ER or whatever. So that's physically how that's happening. And then the NLS is a little bit different. You need more proteins, kind of more moving parts, particularly important and exportant for moving it out of the nucleus. And we are going to talk more about that. And yeah, just also know that when it's entering the nucleus, it's already fully folded. So it would be like that diagram that we showed earlier of the mRNA being translated by the ribosome in the cytosol and then being bound and moving through the nuclear pore complex. So the way that that works, because we just showed that the nuclear localization sequence is kind of in the middle of your amino acid sequence, like in the middle of, you know, your protein. When it folds, that segment, even though it's in the middle of your amino acid sequence, it's kind of going to contort to be facing on the surface of your protein. So your other proteins, your recognition proteins, important, particularly can find that and move it. Does that make sense? Even though it's in the middle of your amino acid sequence, it's going to be on the outside of the protein.
So cells with proteins with no signal sequences will by default stay in the cytosol where they're made. And for localization, ER, mitochondria, or nucleus, the protein has to traverse through a channel, though not unfolded, that's typo. It just depends on which one it is. Yeah, so the only ones that will go through fully folded is nuclear, whereas mitochondria and ER go through single file. And so that just means that like, just to connect it back to previous units, like, it just means that it only has its like primary structure. It doesn't have like second, tertiary, or quaternary just yet. And for the nuclear proteins going in fully folded, it says the nuclear pores are big enough to let them through. And then, yeah, so ER and mitochondrial localization sequences are going to be cleaved off after they are brought to their end destination provider. And so these diagrams just show, this is a nuclear import-export diagram, different than shown later and shown in your videos. And then this one is mitochondrial transport. And so it just shows this protein going in single file.
A lot of flow charts. Okay, so if you're a visual person, this might be good. If this doesn't resonate with you, like, don't even worry about it. But this kind of just is showing, I guess, the paths that you can take from your ribosome to your ultimate protein destination, depending on, you know, what sequence it has. So obviously, like, if you have your ER localization sequence, you're going to go to the ER, and that's going to be on a docked ribosome on your ER that's going to thread it through. And if you have a retention signal sequence at your C terminus, then you're going to stay somewhere in the endomembrane system, but otherwise you're going to move to the Golgi, you're going to be put into a transport vesicle. And the way that vesicles work is they're kind of like flexible little bubbles that can bud off of an organelle and then fuse with the next organelle and then bud off. And it's just like bits of the membrane pinching off and enclosing their contents. So the vesicles will just keep kind of budding and fusing and dropping their contents onto the next stage until eventually it fuses with the plasma membrane and dumps outside of the cell. Then mitochondrial localization sequence, pretty straightforward, same thing. Nuclear localization, we're going to talk, that has like its own 2 or 3 slides. And then for the cytosol, I think we've said this a couple times now, but your ribosomes are in the cytosol, right? So you're coming out of the nucleus with your mRNA, and then it's binding to the ribosome, it's translating, and if it has no localization sequence, nowhere to be sent to, then it will just stay where it was named. So, yeah.
Yeah, so this slide just shows again the picture I showed before of mitochondria import. And then this one's just like a more detailed ER localization chart. And so it just shows the binding of SRP to this ER localization sequence, which in the diagram is labeled as signal sequence, ER signal sequence. And so on your exam, though, you definitely would want to mention that it's ER localization sequence or import, just because there's ER localization and ER retention signal sequences. So, yeah, this is just showing the binding of that co-translationally, and then bringing it over so that it can continue translating into the
Okay. So I kind of just touched on this, but we have a practice question. So if you're watching on Zoom, feel free to like leave your answer in the chat. We can look at it or not, whatever you're comfortable with. But the question says, transmembrane proteins must have ER import signal sequences on their end terminus. Predict where you would find a membrane receptor protein if you engineered its gene such that you remove the ER signal sequence, and then assume that your receptor protein is not completely misfolded or get degraded, because, you know, whenever you're changing the chemistry, the amino acid sequence of the protein, that is possible. But where would your receptor It's okay if it wasn't.
I mean, okay.
Okay.
I feel like the nucleus, because I know the nuclear, the NLS is further into the amino acids.
Right, but that's assuming that the DNA, the gene for this protein had the NLS encoded. So you aren't gonna have every signal sequence in every protein, which I think is important to clarify. We talk about that sometimes as a hypothetical, but generally, like the way that your protein is gonna be coded is just to have the signal sequence for where it's meant to go. Does that make sense?
Saying this.
Right, yeah,
correct.
yep.
Yeah.
So you're gonna stay in the cytosol for anybody that didn't hear on Zoom, because that's where your protein was made. So if it doesn't have a signal sequence, that's where it will stay.
Yeah, so, also like another good hypothetical is, another hypothetical to think about, or like other ones to think about is like, if there's like multiple signal sequences. So like, if there was, let's say there's a ER localization sequence, right? So it means it's going to be, that one means like it's going to be sent to the ER, but it also has nuclear localization sequence and the mitochondrial. Right? So if it has all three, where do you guys think it's going to go?
So it has nuclear, mitochondrial, and
ER localization,
like it could go everywhere.
right? It's like it's like, because it's a lot to like take in. So the important thing is to like keep in mind like the order of when these things are being like read. Maybe, but it's okay. I'm just gonna go back to this one.
What do you mean by one?
So all these signals, like when these proteins have these signals in their in like their codon, like in the amino acids, right?
Right.
They're read at different points in time in terms of translation, right?
Oh,
So it's really important to keep that in mind, because like this comes up on exams a lot, in terms of like when you have multiple, you have to keep in mind like the big picture of what's going on here. So this, like the very first signal to be read out of all of them is ER localization, because it's read during translation, right? Like once, if it has a mitochondrial sequence and an ER localization, what happens is that the ER localization sequence is recognized during translation. So before that mitochondrial one can even be recognized by the appropriate protein in the cytosol, it's going to get moved to the ER, right? And it's going to be translated into the ER. So by the time it's finished translating, it's going to be, when it's a fully formed protein, it's going to be in the ER. And the proteins that recognize the mitochondrial sequence or the nuclear one don't exist in the ER. They only exist in the cytosol, right? So they're not going to be able to bind to the proteins to bring them over to these other places. So this one is automatically going to win out.
oh, so that means it doesn't even go to.
No,
Oh, okay.
exactly. So when you get questions like that, it's good to keep in mind like when these are happening, right? Like, for example, let's say you have no ER localization, but you have an ER retention or a Golgi retention, but you also have a mitochondria, let's say, right? The protein is never going to be, the protein with those signal sequences is never going to have molecules bind to it to have it stay in the endomembrane system, because it never entered in the first place, right? So it's just going to go to the mitochondria. Does that make sense? Yeah.
Oh, okay.
Yeah, I'm saying, sorry, I should have used that.
Sorry.
Yeah. In a case where you have like a bunch of signal sequences, but no localization for ER, right? These aren't going to, no matter what retentions you have, if it doesn't end up in the endomembrane system in the first place, it's not going to be able to be retained in any way.
Right.
That's another thing to keep in mind. So it's good to keep in mind like the order of when these things are happening.
Okay, so what if, say, for example, I have ER and then nucleus, it would first go through the ER,
ER what? Retention or localization?
like localization.
Wait,
What is it?
localization is like being moved from the cytosol to the ER.
Oh,
Retention is like, you've already had that, it's going through the endomembrane system, but in order to prevent it from continuing to go on, the retention. will keep
so
it.
we would say maybe localization then?
Yeah, if it has localization and nucleus, it's gonna end up going to the ER.
Yeah.
And then the end result, since it has, you didn't mention any retention one, so it's gonna end up outside the cell
Right,
by default.
and then would it go to the nucleus after?
No, it
No.
just ends up outside the cell.
Okay, that's,
Done.
okay. I'll ask you.
Because we talked about earlier with your nuclear localization sequence, it's in the middle of your amino acid sequence, right? So when your protein folds, it's kind of just going to get covered up. So even if there were important, like it wouldn't bind, it doesn't have it in the ER
Right,
anyway.
it's
So
okay.
yeah, like
Yeah, because the proteins that recognize those signal sequences only exist in certain places in the cell
Okay,
where they're needed.
so just say, sorry, I'm just like,
Mhm.
it just say there was like a Golgi single sequence and then mitochondria.
Mhm.
It would need an ER localization to even go to
Yeah,
Golgi, so it would
exactly.
immediately go to mitochondria.
Exactly.
Okay.
Perfect.
yeah, so there isn't like a specific Golgi localization sequence because it has to go
Yeah.
to the ER
But
first,
yeah,
and then it'll go through to the. But
Yeah.
otherwise, yeah.
Right, okay.
go ahead to
There, okay. Here. Do you want
you
to,
told me, I
oh,
was
okay, right.
confused.
Okay, so we talked a little bit about insulin in class, so this would kind of be an example like applicable to that. But this is like more what I was talking about earlier about how vesicles travel through the endomembrane system. So if we have, like, this is our mRNA coding for insulin. Insulin is a protein. So it's going to dock on our ER. It's going to thread our amino acid sequence through. Then we're going to have enzymes inside the ER that will process that protein as it's being made and after it's been made. So we're going to have an enzyme to cleave your signal sequence. And then you have your prohormone, which is like your not mature, not biologically active version of insulin that still needs to be processed a little bit. So it's going to keep going through your ER. And then this is what I was talking about. Your membrane is going to pinch right here once you have your prohormone insulin in there, and it's going to form a bubble. And then that bubble is going to fuse with this membrane, and it's going to go through the entire Golgi complex like that. I think like the analogy that usually people use is like stacked plates or a pile of pancakes. It's kind of just like all of these long bubbles. So it's going to bud and fuse all of the way through the Golgi apparatus. And then, assuming that we have no retention sequence, which with insulin we probably wouldn't, because we would want to send it out into the body, we're going to have a little bit more processing. So, you know, our active hormone is going to be cleaved and altered whatever way that we need it to be, then packaged up and put in a little secretory vesicle. And then this is our very outside plasma membrane. So it's going to fuse with that last plasma membrane and be put, this is the extracellular fluid. So then it can diffuse or be, you know, transported, probably active transport somehow, into your capillary and then like go throughout the body. So essentially just with this, remember your vesicles are also a part of the endomembrane system, and remember that they are transporting your contents, your proteins, whatever, just by budding and fusing with, you know, the organelles in that system.
Yeah.
And that's called exocytosis, by the way, when it spits them out.
And since like it's being excreted, like the cell, you just secrete from the cell, you only need ER localization. Because then,
Yeah.
by default, it gets secreted.
It critically secreted if it happens.
Yeah.
Which we want.
So lysosomal enzyme, lysosomes are involved with waste removal of products of the cell. And so they will travel through the endomembrane system. And after they go into the Golgi body, will be put into a vesicle and then stay in the cell for waste to be sent to them in order to get rid of. It just causes like it breaks down the particles like inside of you. Yeah, I'm just watching.
So,
You don't have to know too much about that.
so lysosomal enzymes have like their own, do they also get like a sequence to go through the, or is that, is this
Yeah,
just like an
but, like,
extra?
Yeah, but you don't need to know too much about that.
Oh, okay. So
Yeah.
what was the main thing? Is that they go through?
Yeah, then they just go through the endomembrane system and then get it sent out from there.
So. You don't have to know a ton of it. I mean, really, okay, so we're gonna talk about RAN because RAN is important for nuclear import and export. RAN is a part of the RAS superfamily. RAS is another protein, very similar relative that you probably talked about in 151 that I think we're gonna talk about in topic 5. But RAN is a GTPase protein in the RAS superfamily. And GTPases basically hydrolyze GTP, which is an active protein, to GDP, but it has a very slow intrinsic rate of hydrolysis. So kind of like we talked about with enzymes, some reactions in the cell will happen naturally, spontaneously, but they'll happen too slowly for them to be biologically valuable because these things that are happening in the cell have to happen really quickly and in really large quantities for them to be valuable and impactful. So that's why we're going to talk about GAPs and GEFs. So just remember that RAN, even though it can hydrolyze GTP to GDP, it has a really slow intrinsic rate of hydrolysis. So it needs a little help sometimes. And that is going to be again important for import and export. And you don't really need to know anything about the RAS superfamily. It just might be helpful to know that they're related, which isn't hard to remember because they sound similar, but they're going to function very similarly, just in different pathways. I don't think that we talk about, no. I think it's
Just sounds like
just
later on at the top.
really other than RAN in that family,
Yeah, like
Oh.
RAN. Wait, sorry, did you already say that?
RAN and RAS.
Oh, yeah, sorry.
Okay, yeah.
So yes, a little bit about GEFs and GAPs. GEFs are guanine nucleotide exchange factors. And so they will facilitate the exchange of GDP to GTP by having by helping a GTPase decrease their affinity for GDP. And so what happens is that it'll bind to a GTPase active site, and then will cause like I said, like the decrease in affinity for that protein to the GDP. So it'll dissociate out the GDP and GTP that is floating nearby in the cytosol will bind to the GTPase or the protein of the GTPase. And then once it does its job, it'll end up unbinding, and then the RAN GTPase will be bound to GTP. On the other hand, GAP does like the exact opposite. It's called GTPase activating protein, and what it does is it increases the rate of hydrolysis of GTP of the GTPase. The rate of hydrolysis of GTP to GDP, so it'll help increase the rate of the reaction of RAN cutting off the third phosphate on one triphosphate. And so it will convert basically RAN GTP to RAN GDP. RAN bound to GTP to RAM bound to GDP.
So nuclear import, I mentioned this, I think before we started, but if Laura does her exam the same way that she has, which I think she will, this diagram will be printed out and given to you. So you don't have to memorize all of the fine detail, and so will the export diagram. You don't have to memorize all of the details, but certainly like be able to interpret this and know what it means when you're looking at it. So this is like the general process for how a protein ends up in the nucleus. So you're starting with your cargo protein. We have our NLS, which is in the middle of your amino acid sequence, but as the protein is folded, it will be put on the exterior surface. That has a binding site for important. So important will bind your cargo, and that's going to enable it to move through the NPC, which stands for nuclear pore complex. And it's going to move in again, fully folded. So then once we're inside, the cargo has to let go of important. So it can go and do its thing, whatever, it needs to unbind. But we talked about KD and affinity. Presumably they have pretty good affinity in this conformation for them to be able to hold on long enough to move through the NPC. So we need something that's going to decrease the affinity between the cargo and important. So that is when we are going to use RAN. So in its like inactive state, RAN will already be in the nucleus in the GDP bound state, and that's going to be inactive. So what we need is GEF, guanine nucleotide exchange factor, to bind to RAN. And when GEF binds to RAN, that's going to change the conformation, the shape of RAN. And RAN's active site is going to change. So it drops GDP because it loses affinity, and it gains affinity for GTP. So GTP is going to bind RAN, and RAN is going to be active. Okay. So then with your active RAN, you can take your cargo and important complex. Those are going to do the same thing. They're going to interact. That is going to cause the cargo and important to lose affinity for each other. So essentially think about any time that you have a chemical interaction taking place, anything binding, that's going to cause a change in shape, and either that will increase affinity or decrease affinity. So when GEF binds to RAN, it loses affinity for GTP, GDP, it gains affinity for GTP. Same thing when RAN binds to your complex, your important cargo will lose their affinity, they'll dissociate, and then your cargo is released. Your RAN GTP can exit with important, and then you have GAP, which speeds up that really slow intrinsic rate of hydrolysis and allows RAN to again inactivate itself and become GDP bound, and then it cleaves that last phosphate and you can go back through that cycle. Does that all make sense? I know that's kind of a lot, but
And I have to
that's totally okay. This took me a really long time.
How does the RAN get?
RAN is just in the nucleus, like, so presumably like at some point there is going to be a gene that codes for RAN, and just like any other protein, it's going to be translated and imported, probably actually with
RAN?
yeah.
Okay.
So I don't like, I guess that's kind of a chicken and egg thing, but RAN just lives in the nucleus. So and also RAN will be in your nucleus. GEF will also be in your nucleus. GAP lives in the cytosol. So this might be something, this last line here might be something that comes up on an exam. And you can kind of see with this and this where they are.
The
But
question you
sometimes
asked won't be on. That's something
yeah,
that she
right,
states.
right,
like,
right.
She like states it's like a cutoff of like, we don't go over that in this class.
Yeah, so you will probably need to know though that, you know, you're at least be able to apply that your GEPs are in the nucleus to make your RAN GTP bound, and then your GAP is in the cytosol. So,
Any other questions on
do you
import?
have a question?
I have a question. No.
Okay. Okay,
Do you have a comment?
No, I just like won't know it just from this. So
that's totally okay. You can also, like, if you want to look at it after and go through it, like, look at this diagram again, you can always come to office hours.
Or like Laura's help sessions, any of that.
Yeah. I mean, I feel like it's simple. I just need to like.
There's a lot of
Yeah.
steps in it. There's a lot of steps in it. A lot of it comes down to like,
You have to memorize the steps.
no. You just gotta understand like the diagram, because like these diagrams will be provided.
Right.
Yeah,
So
so
what was,
Like know
No, I
what
think
RAN
it's-
is, like know what the cargo protein does, like
Yeah.
know what, okay.
Yeah.
like looking at this, just know how to
what
interpret
it
it and
works
say
for.
like, oh, when you're looking at like step 3, oh, well, you know,
Right,
Geth
what's
is interacting, yeah, right.
Right, okay.
And then like if you're asked a question, like, oh, what would happen if you didn't have Geth? Like be able to visually just cross that out and say, okay, well, if we miss that step, then what would the impact be? So
Okay,
like, don't worry about that yet, just get comfortable with how it works.
okay,
And then expect that you're not gonna be asked to regurgitate it, you're probably going to apply it.
right.
So, and we can talk about that also in office.
yeah, and a lot of this just like comes down to like, a lot of these, understanding a lot of these steps comes down to like understanding the core concept of like, what is the gap doing, what is the gap doing? And then also, a lot of it comes down to like a protein binding to another protein, causing some sort of conformational change that causes like some sort of dissociation or a reaction, and then somebody else dissociates. It's a lot of proteins binding to proteins causing conformational changes that cause something to happen. And the other thing is that like there's nuclear import and export, but the diagrams for both of them are pretty similar. So like the GEF and the GAP part are really similar. Yeah.
Make sure you read the label on the exam. If you
Yeah.
Don't mix them up.
Definitely don't worry. Yeah.
But don't do it, 'cause they will be in black and white. So like, I think the other one is purple and this one is
Yeah.
green, the cargo or maybe yellow. So it will be in black and white,
Okay.
so read the label,
Okay,
especially if you're not super comfortable with it.
right, okay.
So the export cycle is pretty similar in terms of like the setup. Where you've got like GAP over here, you've got like RAN working here, you got GEF up there. There's just a few differences. So the start of this process is this cargo protein right here needs to start like way up here instead of starting down here, right? Where it's with import, you're importing to the nucleus, so obviously the protein starts outside. For this one, we're starting up there with the cargo protein having an NES sequence. And so what's going to happen is that when RAN is in the GTP bound state, after it interacts with GEF, then it's going to be in a conformational state where it's going to be binding to exportin, and then exportin will then change conformation when bound to RAN GTP. And then in that conformation, it's going to have high affinity for the NES sequence on the cargo protein. And then all of that complex is going to come out of the cell, the nucleus, not the cell. This is all within the cell. And then once it comes out of the nucleus, it's in the cytosol, and its job is done. Like this whole complex has completed its task of having the cargo in the cytosol exported from the nucleus, but it needs to be free. So in order to do that, GAP is going to come in and it's going to convert the GTP, the RAN GTP to RAN GDP by doing its thing as usual, binding the active site, helping it speed up its intrinsic rate, because it already has a rate of, it already can hydrolyze GTP on its own to GDP. It's just very slow. So this speeds it up, helps convert to GDP. And once it's in this inactive GDP state, it's going to dissociate from exportin, and then exportin is going to also change conformation. It's going to no longer have affinity for the cargo. The cargo is going to be released. And then this allows, this frees up not only the cargo, but it frees up exportin to be able to go back into the nucleus in order to repeat its job. So like a big example she likes to give out is like, or yeah, it's like what Lily was saying earlier, where you take out GAP or you take out GAP. Like if you take out GAP here, a lot of these revolve around like, where's exportin going to be? Is there any available to do this job? So like if you were to get rid of like GAP, right? This is going to start to build up outside of the cell, right? With like RAN GTP being bound to exportin, bound to the cargo, and it's not going to free up the exportin, and then it's going to end up having all of its exportin outside of the nucleus, and it no longer will be able to send proteins outside of the nucleus to be exported.
At any point before the exam, will you like do possible like little practice?
We have exam reviews, and I think for the most part, we do practice questions, or at least we include practice questions.
Okay.
But come to the exam reviews, they're helpful.
Yeah, we really want Kahoots for that.
Yeah.
Sorry?
Kahoots.
Mhm.
Yeah, like
Like
the game.
things go wrong?
No,
It's like-
like
it's
Kahoot
like
the
a-
game.
It's
Oh.
a game.
Wait, sorry. You know Kahoots. Oh, okay. So it's a game. It's just like a game where basically like.
It's almost like iClicker, but ranked. It's like ranked iClicker.
Oh,
Yeah.
okay.
Yeah.
Oh, I guess that's a good better way to explain it. I was
Yeah.
gonna explain like multiple choice and
Okay.
stuff, but
Yeah,
it's just
so-
it's basically.
Like you log in and like you put your name or like something funny, and then there's a thing up on the board and like a 30-second timer, and you click the answer, and then like based on whether you get it correct or like how fast you answer it, it will like rank you, and
Okay.
Yeah,
Okay.
but then we like review the questions, like just like in
Right.
class, like
Right.
we'll review them.
it's fun.
But she doesn't have any multiple choice, does she?
No, but
This is
these,
the final.
yeah, the
Yeah.
final usually does.
So she said possibly this semester there might be multiple choice, but I would not count on that.
Yeah. But like a
For
lot
sure,
of
don't
what we're
count on
doing is like, then you think about it, and like
Yeah.
the reasoning that we'll walk you through will be like what you would put on the exam.
Right, right.
And then those
right.
exam reviews will be. So,
Right, okay,
you'll-
because that's what I'm wondering, like, like questions like this, like, okay, what happens if this is not there? Like, I need to like sit down for the briefing and like write those answers down.
yeah,
And also like your problem sets. I think she's still at the end of the week or like after you turn them in, we'll post the answer keys. Read those.
Oh,
Yeah.
okay.
I think she's still. Is she
Yeah.
doing that? Okay,
I
yeah,
think so.
yeah.
So those questions are like exam.
Yes, so think about problem set questions being exactly like exam questions.
The other thing to do is like, what typically happens is like, you'll see these questions, multiple choice questions on, your assessment quizzes or whatnot, those quizzes online, right? And then those may or may not be converted to
Right,
response
right,
forms.
questions, right. We just need to collect all the questions.
Yeah, well, so you can collect all the information too on your cheat sheet, so just make sure that you understand all, like that is really, just if you have a question, like, don't linger on it, clear it up, and you'll be fine.
Right.
Like,
Okay.
so it is 8 o'clock. I don't know if any of you guys have to leave.
No.
Okay. Okay, yeah, we have some more slides on like
We
processing.
don't have too many left.
No, we have, and some of them we can go through.
Where are we? Oh, we're at 23.
Yeah, we. Some of them we can go through pretty well, though. Like that one will take long.
Yeah, okay.
We'll go through we're gonna post this, we'll go through the last ones kind of quick, and if you guys have questions, like email us, take it to office hours, but don't feel pressured to stay, like if you have to go. We usually try to run full count, so, okay. I think we might have missed one.
What? Oh.
Slides. Yeah, okay. All right, so, one of the things that regulates transcription in the cell is transcription factors. Your general transcription factors are just going to be the ones that help your RNA polymerase binds to your DNA promoter region to like initiate your transcription. And then you can have regulatory transcription factors. There's some ones called specific transcription factors that are going to only work and like bind and apply to specific proteins.
So your positive ones are enhancers, they're gonna increase transcription, and then your negative ones are silencers. So if you have a negative transcription bound, then you're not going to have transcription when it's bound to that DNA. So does that make sense? Right, and that's just a level of control. So like if you don't want a protein to be translated, one location that you could do that at is like by having a negative regulatory transcription factor to prevent it from even being transcribed, like before translation is even a thought. So this is just a type of like regulation.
Another type of regulation at the transcription level is whether you have so DNA is like wound up around these proteins called histones. And when this altogether is called chromatin. It has two forms. One is open, where it's like unwound. These are I made some examples. When it's open, it's able to be read by all those transcription factors we were talking about. When it's closed, it's going to not be able to be read by the transcription factors. It's pretty funny to know for that part.
All right, so RNA, before it is sent out of the nucleus, also has to be modified to make it stable. So there are going to be two main methods that we do for that. One is the 5' cap, and essentially, like you have nucleases that will chew at the ends of your mRNA or your RNA. You don't want that to happen because then it will be degraded before it gets to be translated by the ribosome, but those just kind of exist in the cell. So your 5' cap prevents that chewing at your 5' end of the protein. And the way that that works, you don't really have to know this in super great detail, but there's a specialized guanine that has a different structure chemically, and it's added almost like upside down. It's added in a different direction. So your nuclease binding sites don't recognize it, and it can't bind to chew that away. And then at the other end, at your 3' end, you're going to have a poly A tail, and that's right here. It's literally just a long sequence of adenines, and those will be chewed over time. So if for whatever reason, like it takes too long to get your mRNA to your ribosome, then your nucleases will degrade it. But there's enough there as a buffer that even as they're chewed off little by little, you should have a safe mRNA by the time that you reach the ribosome. And this is necessary for export. It's kind of like a signal it's ready to go, process, whatever. So.
Another thing you can do is RNA modification and processing. So after a piece of RNA is translated from DNA directly, not all of these will make it through to code for the final protein. Introns are pieces that will be cut out. Exons will be pieces that will stay in. And then spliceosome cuts out these introns, splices together these pieces of these exons together, and then those will compose of the mature mRNA, which will then be transported outside of the nucleus to be translated like with the cytosol. And again, this is all happening in the nucleus. The important thing to note here, like how to keep these two straight, because like they seem like they're the opposite, is that introns are in the way, so they need to be cut out. Exons don't have anything.
Extra important. They're extra important. All right. So in addition to that, you also have alternative splicing, which is really important because think like you have 20 amino acids to make, and like 20,000 to 25,000 genes to make possibly hundreds of thousands of proteins. I saw an estimate that was like, you know, the human genome can code from anywhere from like 85,000 to 400,000 proteins. So you need a lot of different ways that you can combine those to make that amount of proteins with your number of genes and amino acids that you're working with. So alternative splicing, you take one gene, you splice, your spliceosome takes out your introns, you have your exons remaining, and then when you do alternative splicing, you get rid of some of your exons. So we, like here we have exon 1, 2, and 3, here we have 1, 2, and 4, and from the same gene, this allows us to create different protein products, which increases the diversity of proteins that you can make with a relatively small amount of protein.
Another couple ways it can be controlled is that mRNA can be degraded. And we'll talk about RNAi, which targets specific mRNA to be degraded. And then also there can be controls on at the translational level, like whether or not a mRNA will be translated into a protein that it codes for. Again, in our RNA interference can play a role in that, but we'll talk about that later.
Okay, so this is just a quick comparison. You guys can also like read this on your own if you want to, but this is like how the eukaryotes versus prokaryotes regulate their transcription. So one of the really important things is prokaryotes don't have histones, so they can't have chromatin. They don't have membrane-bound organelles, so you're not gonna have like processing, RNA processing in the nucleus, because you also have transcription and translation occurring at the same time. They have different general transcription factors. It's called the basal transcription factor for eukaryotes, the sigma factor, which we're going to talk more about in prokaryotes. And then these are all kind of just things that they do the same. So, you know, these you can just read those on your own, but we'll keep moving.
Yeah, this is just a general overview of what we were just talking about earlier, where but just in a row, right? So like all this nucleus, all this cytosol. So like, right, this is like transcriptional control, whether or not it's transcribed, DNA to RNA. This is like the exon, intron stuff we were talking about before. And then also this can be degraded. Oh, that's right. And then outside in the cytosol, once the mRNA makes that to the cytosol where it will be translated, maybe it has to be transported and localized, whatever needs to be brought out to here. It has to be translated. It could not be translated. And then it also can be degraded, like I said. And then once it's been translated into a protein, it can be controlled by, its expression can be controlled by degrading the protein or modifying it or inactivating it.
All right, I think this might be our last slide. So this is the same diagram, but for prokaryotic regulation. And you can see here, like some of the steps just don't happen. The trend, and you don't need to know this in as great detail as you do for eukaryotes, but the trend that you want to remember for prokaryotic gene regulation is like you don't have your membrane-bound organelles, you don't have chromatin. So like there are fewer locations where regulation can happen, but you can still regulate like your levels and rates of things. So translation rate, like that's not dependent on having membrane-bound organelles. Yeah, rate of degradation, rate of transcription, like all of that is still going to happen in prokaryotes. But if you ever were to get a question about this, rather than memorizing every single thing that prokaryotes do and every single thing that they don't, just think about like the way that a prokaryotic cell is structured. Would it even be possible? Like, could they regulate with chromatin? No, because they don't have histone proteins, that kind of thing. Like, could they regulate or process their mRNA? No, because translation is going to be happening at the same time as transcription because they both happen in the cytosol. So that's kind of the takeaway.
Also, you can just draw this on your cheat sheet.
True, yeah.
Thank you for coming.
All right.
Questions? Any questions?
If you do, we're gonna post this, especially if our audio is weird, so you know, come to office hours, go to Laura's help session.
Thanks for asking questions.
Yes, yeah, your questions were fantastic.
Yeah, tomorrow mine's 4 to 5.
Yours is 45,
Yeah.
and then
12:30 to 1:30 on Fridays.
is yours.
Yeah.
And then Laura's help sessions are.
2 to 3, I think, on Fridays.
Yeah.
And it's just like
Or 2:30 to
sessions where we just go and.
It's like extra help, like
Yeah.
you can, usually, especially for the Friday one, because people procrastinate, like for my help session, we mostly do problem set.
Okay.
But if you have other questions, you can literally come with anything. And if you're struggling with your problem set,
Like I guess